Discoveries from many different branches of science, including evolutionary biology, paleontology, and microbiology, have come together to form hypotheses about the origin of life. The scientific evidence collected from decades of study shows that life originated 3.5 to 4 billion years ago from non-living matter in a series of 4 stages. The 4 stages that explain how life originated from non-living matter are Stage 1, organic monomers, Stage 2, organic polymers, Stage 3, protocells, and Stage 4, living cells. In Stage 1, organic monomers were created. The first ever organic molecules in simple forms originated from inorganic compounds that were present in the Earth's early atmosphere before cells were even in existence. Examples of the simple organic molecules that formed during Stage 1 include amino acids, nucleotides, ATP, sugar, and lipids. In the 1920s, 2 scientists, Alexander Oparin and J.B.S. Haldane, each proposed that the first stage in the origins of life was the creation of monomers from the inorganic molecules that were present in the Earth's early atmosphere. The early Earth had methane and ammonia in its atmosphere. These reducing agents easily donate their electrons in the absence of oxygen. Therefore, oxidation and reduction reactions likely drove the abiotic synthesis of the organic monomers from the inorganic molecules. In Stage 2, organic polymers were created. These organic polymers were formed from the joining of polymerizing of organic monomers in the cell's cytoplasm in the presence of enzymes. Examples of organic polymers include DNA, RNA, and proteins. Sidney Fox created the protein first hypothesis, which proposes that protein enzymes were formed before the first DNA molecule. Other scientists believe that only RNA was present before the formation of the first cell. In Stage 3, protocells were formed. Moving from the organic polymers in Stage 2 to the protocells of Stage 3 transitions from chemical evolution to biological evolution. Organic polymers became enclosed in a plasma membrane to form protocells, also known as probionts, which were the first cell precursors. The formation of the plasma membrane was needed for the maintenance and regulation of cellular function. The plasma membrane serves the critical role of separating the inside of the cell from the outside world. Although today's cell membranes are made of phospholipid bilayers, the first plasma membranes were likely made of fatty acids instead. In Stage 4, the first living cells are formed. Protocells undergo biological evolution and produce the first true living cells that have the ability to self-replicate and perform other cellular properties. There are many theories about the order of creation of DNA, RNA, and protein synthesis. It is possible that the sequence developed in stages. There is also evidence that the genetic code underwent natural selection before evolving to today's code. Fossils, which are the remains and trace of past life, document the history of macroevolution over long periods of time. In the early 19th century, scientists began the process of relative dating of fossils. Geologists began to correlate the strata from around the world of the same age. They identified index fossils that would serve as a reference point to allow them to identify deposits that were made at the same time around the world. The index fossils were used in a process called relative dating that allows them to identify the ages of the strata from around the world. In addition to relative dating, absolute dating is also performed, which uses a radiometric technique to generate an actual data for a fossil. Radiocarbon dating uses the radioactive decay of carbon-14, which is a rare carbon isotope, to date fossils. Geologists have studied all of the fossil data from strata around the world to create the geologic timescale, which divides the history of the Earth into areas, periods, and epochs. The geologic timescale begins with the Precambrian era, which is when life began with the creation of the first cells. This time period accounts for 87% of the entire geologic timescale. This time period covers the period of time between 4.5 billion and 630 million years ago. The Precambrian era began with Earth's formation 4.5 billion years ago. Single-celled prokaryotes, which were the first modern-type cells, next evolved during this time period, 3.5 billion years ago. Atmospheric oxygen began to accumulate 2.7 billion years ago, which allowed organisms to live on land. The first eukaryotic cells evolved 2.1 billion years ago and obtained energy from cellular metabolism in the presence of oxygen. Protists began to diversify 1 billion years ago. Soft bodies and vertebrates were created 630 million years ago. During the Precambrian era, the first cells evolved to bacteria and archaea. The first eukaryotic cells evolved from the archaea. Eukaryotic cells gained mitochondria by engulfing aerobic bacteria, which led to the creation of heterotrophic protists. Photosynthetic protists formed when these cells gained chloroplasts after engulfing photosynthetic bacteria. Animals evolved from heterotrophic protists, and plants evolved from photosynthetic protists. The Paleozoic era lasted for 300 million years, which was much shorter than the Precambrian period. It was divided into 4 periods: the Cambrian period, the Ordovician period, the Silurian period, and the Devonian period. Mass extinctions occurred in which a large number of species disappeared. Over 57% of marine species became extinct after the Ordovician period. Over 50% of coastal marine species and corals became extinct after the Devonian period. During the Cambrian period, all marine algae flourished. All animals can trace their ancestry to this time. Life moved out of the ocean and onto land. All invertebrate phyla and their first chordates appeared. During the Ordovician period, non-vascular land plants appear on land. First jawless and then jawed fishes appear. During the Silurian period, seedless vascular plants appear. Jawed fishes diversify and dominate the seas. During the Devonian period, first seed plants appear. Seedless vascular plants diversify, and the first insects and first amphibians appear on land. The Mesozoic era lasted for 294 million years. It was divided into 5 periods: the Carboniferous period, the Permian period, the Triassic period, the Jurassic period, and the Cretaceous period. There were 5 periods of the Mesozoic era. The first period was the Carboniferous period. It was the age of great coal-forming forests comprised of ferns, club mosses, and horsetails. Amphibians began to diversify, and the first reptiles appear. It was the first great radiation of insects. The second period was the Permian period. Germanospores diversify, and reptiles diversify, and amphibians begin to decline. During the Triassic period, there was a domination of forests that included conifers and cycads. The first mammals and dinosaurs appeared. Corals and mollusks dominated the seas. During the Jurassic period, flowering plants appeared. Dinosaurs flourished, and the first birds appeared. During the Cretaceous period, there was a spread of flowering plants, and placental mammals and modern insects appeared. The Cenozoic era lasted for 65.5 million years. It was divided into 2 periods, the Tertiary period and the Quaternary period. Significant mammalian extinction happened after the Quaternary period. The Tertiary period of the Cenozoic era is further divided into 3 epochs. The first is the Paleocene epoch, in which flowering plants diversified and primitive primates, herbivores, carnivores, and insectivores appeared. The second is the Eocene epoch, in which subtropical forests were sustained by heavy rainfall, and all modern orders of mammals became in existence. The third is the Eocene epoch, in which many modern flowering plants families evolved, and browsing mammals and monkey-like primates appeared. The Quaternary period of the Cenozoic era was further divided into 3 epochs. The Mesolacine epoch was when grasslands spread and ape-like mammals, grazing mammals, and insects flourished. The Pliocene epoch was when herbivorous angiosperms flourished and the first hominids appeared. The Pliocene epoch was when herbivorous plants spread and diversified, and Ice Age mammals and modern humans appeared. The Holocene epoch was when humans began to influence plant life, known as the age of the Homo sapiens. How do the Catholic beliefs link to the theories on the origins of life and geologic timescale? Evolution has consistently been used as an argument against the Catholic Church teaching that God created the universe and man. Pope John Paul II acknowledged the scientific evidence behind evolution and tried to make the connection between the Catholic religion and evolution. The Book of Genesis teaches Catholics that God created the universe in 6 days, which is a yurta. The process of evolution, from which man was created from a single-cell organism over billions of years, could be the very process that was controlled by God to create the universe and everything in it. What is most important to Catholics is the belief that God was behind the creation. Scientists can't explain exactly how inorganic material was transformed ultimately into man. The Bible tells us that God formed the man of dust from the ground. The Catholic belief is that this transformation was done by God. Finally, the other critical premise that the Catholic Church overlays onto science is that God created the soul of man, which is a belief that cannot be compromised by science. Regardless of religious belief, scientific research over the past century has shaped into the origins of life and the geological timescale that have become the widely accepted theories on evolution.